ठीक शुरू. Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and this is our third and the final live session for the experimental biotechnology. So if you have any concern and queries, queries related to the topics or queries related to the exam or other kinds of queries, please you can send your questions and I'll be happy to help you. Good afternoon, Nidhin. Do you have any questions? So both Nidhin and Abhishek is asking the similar questions. So we are going to have 100 questions in the exam, and every question is going to carry the 1 mark. So there will be 100-mark questions. Sai is asking about how the vacuum transfer of DNA is working for the nitrocellulose membrane. So you can imagine that the vacuum transfer or the capillary transfer method is exactly the identical, except that in capillary transfer method you are actually using the uh, suction pressure. You are using the suction pressure which is being created by the capillary action of the water from the buffer to the nitrocellulose, the to the blotting sheets. And whereas in the vacuum transfer method, you are actually applying a vacuum across the nitrocellulose membrane. So because the vacuum, when you are externally applying the vacuum, it is sucking the water from the below reservoir, and in that process it is also sucking the DNA. So when it is sucking the DNA, because the nitrocellulose membrane is not going to allow uh, the transfer of the or the pass through of the DNA through the nitrocellulose membrane, that DNA is going to be remain stuck onto the nitrocellulose membrane. And because the capillary action is having a very slow suction pressure, that's why you require 16 to 20 hours of capillary transfer, whereas in the case of vacuum transfer, because the vacuum transfer is going to be very fast or very powerful, because you're externally applying a vacuum, it is going to take much lesser time. But ideally, as far as the procedure is concerned, you're using the different types of instruments, but the principle remains the same. In one case, you are using the suction pressure of the vacuum capillary action. In the other case, you are literally applying the suction pressure with the help of the vacuum pump. Sai, if you have any more questions, you can ask me, because I think you were asking me later many questions, and few of the questions I could not understand because of the either you could not explain the things in detail, or there would be some language issues and so on. So feel free to ask me. I am. It's going to help you in solving the questions when in the final exam also. I think this course, or probably I should not give you any answer or queries, responses against the queries which are more related to the IIT versus, which is more related to IITs. So there are, set of people who are responsible for conducting the entrance exams for IIT, whether it is JEE or GATE, and I think they are the best person to guide you in terms of giving you the proper informations and the complete informations. So better you should raise your queries or questions to them. This live session is very limited, only for 1 hour, so I think this 1 hour you should utilize only for asking the question which is relevant to the experimental biotechnology, rather than how the entrance exam works in IIT and all that. इस साइड का बड़ा कर सकते हो? So Sai is again asking, how many ways we can lyse a cell generally in a basic laboratory. So, as far as the lysis of a cell is concerned, there are multiple methods and different types of methods which anybody can use, uh, and it depends on the physical as well as the particular requirement of a particular procedures. For example, I think we discussed little bit briefly about lysis of the bacterial cell as well as the mammalian cells. But see, imagine if I have to isolate a protein from mitochondria, right? Then what we have to do, and suppose it is from the animal cell. So what we have to do is we have to be very gentle in terms of the breaking open the plasma membrane and then isolating the mitochondria and then breaking the mitochondria to isolate that particular protein. But in this process, we could use some milder methods so that we should not harm the mitochondria while we are lysing the plasma membrane and trying to isolate the mitochondria. So, considering all these factors and the requirements and the sensitivity of the factor which you are going to isolate from the cell, the cell lysis method could be of three different types. Either it could be a physical method, it could be a mechanical method, or it could be enzymatic method. Classical example of the physical method is that you can use some detergent, or no, physical method you can use some physical parameters. For example, you can use the thermolysis, or you can use the osmolysis, and so on. But as I said, if you use this method, probably you will not be able to isolate the mitochondria, because if you do osmotic shock to the cell, then the mitochondria also is going to lyse. So if you try to use the osmotic lysis method or osmolysis, then probably that may not be good. Similarly, if you use thermolysis, so in that case, what people do is they just heat the cells, and while when they heat, the cell expand actually, and how much they can expand. So it actually goes to the lysed later on. But that also depends whether your product what you're isolating is thermostable or not. Because if it is not thermostable, then it is going to be of no use. Similarly, you have the some of the physical methods, like you can use the sonications, or you can use the homogenizations, and so on. And then we have the mechanical method, like where you can use the shear stress and other kinds of parameters. So considering the susceptibility of the factor which you are trying to isolate, and considering where it is present, you can actually have the different choices to be used. I hope you understand what we just discussed. If you did not understand, or you want to elaborate me some more aspects, we can discuss that also. So Vinita is asking about the results and certificate related to the exam. Probably we should consult the NPTEL office in IIT Madras, and you can reach to them by nptel@iitg.ac.in, and you should raise your queries, and they will be happy to help you. Um, uh, then I think the Nidhin is asking about the coupling of the receptor covalent bond is formed. In that case, column could not be used further. Yeah, if you are trying to use the coupling of the receptor with a ligand or receptor to the column, then that column is going to be designated only for that particular antigen or the antibody to be purified. So it cannot be used for some other antigen or other antibody, but it can be used on multiple occasions for isolating the same amount. Because as you might have, I think, as you might have seen, that when we were coupling the antibodies or to the antigen to the receptor, we are not using any destructive method, so that it is actually going to destroy the column, or it is actually going to have any such problem. So it's not a covalent chromatography. Even affinity chromatography is also a non-covalent chromatography, because the interaction of the antigen antibodies are mostly being governed by the non-covalent interactions, like hydrogen bonding, salt bridge, electrostatic interactions, or even the hydrophobic interactions. So these interactions can be broken down, and that may not destroy the column. So you can actually reuse the column for that particular antigen, but not for other antigens. So Vinita, I think you can write a mail to this particular email ID, support@nptel.iitm.ac.in, and I think the people at IIT Madras probably will help you in terms of your queries and any kind of questions what you have related to certificate or exams, or exam centers and all that, because they will be happy to help you. So, Shalini, whatever the calculation you have done is perfectly fine, but all this is based on a probability. So, 4% error we are assuming if it's incorporating, you know, if it's incorporating the, you know, one in every 10,000 basis. So, uh, but what is more concerned about, when we doing the PCR, whatever you have calculated is perfectly fine. But what is the major issue is that you don't know uh, where this mismatch or misbase is going to be incorporated. So if it is not been incorporated into, or it's not been done at a place where it is going to harm the enzymatic activity or the function of that particular protein or the gene, then people actually accept that particular amplified sequence. Otherwise, what you're talking about is probably via data for tag DNA polymerase, and then in that case, what people do is they just go with the more high fidelity enzymes, and then in that case, the error rate is going to be even reduced. And but even then, PCR is never going to be error free. It is definitely going to incorporate the errors, and that's why the biological system has a proofreading activity, and that actually corrects the errors and whatever whatever the enzyme is incorporating, and that's how uh, the mismatch or the base incorporation error is very low into the biological system compared to the in vitro PCR based systems. So exam will be conducted online on the computers. I think you will be assigned a particular center, and they will issue an admit card, and then probably you might have to go there and attempt the exams. Just like as we do for GATE and JE exams. So it's going to be an online exam, but you have to go to a particular center. It's going to be so in the case of experimental biotechnology, it is going to be objective questions where you are going to have the 100 questions, and each question is going to carry 1 mark, and out of those four options you are going to have one option which is correct. So um, so I think, Kushi, I think I have answered your questions. And Renji is also asking the similar kind of questions. So I think there are 100 questions which are going to be of MCQ type. So in genetic engineering, the other course also, Khushi, the pattern is remain the same. Pattern is also going to be identical. You're going to have 100 questions, and every question is going to carry 1 mark. So, but it's better to come for a live session, into the other course also, and then if you have any queries, you can ask the questions there also. So there will be no negative mark for the exam. You can attempt all the 100 questions, and you will get marks only for the correct answer. So there will be no negative marking. So Shalini is, I think the purpose of this course is only to tell you the how to design the experiments and how to execute that. And I'm happy that our course is helping you in advancing your research or teaching activities. And you should you should take more courses from the MOOCs platform and at least increase your knowledge base so that you will be able to more confident and your better you will be able to explain the things in to the your students. I think if you have any queries related to this certificate and other things, probably you should contact the IIT Madras guys, because I think the guidelines are pretty set. And I think you might have read about the guidelines, under what guidelines you will get a certificate or not. So I think better you write to them and they will be able to help you a much better way than I will give you some information and that may not be complete. So Kushi, I think you should contact uh, the IIT Madras NPTEL office and they will be able to help you. Uh, Again, I think you are asking something which is beyond this course. So Bhagirath is asking because of probably his more interest in Corona, but I am not a very big expert of the virology and all those kind of field. But uh, as far as the uh, these tests, what you said is RT-PCR or rapid antigen test or CT scan. All these tests are actually providing the different types of informations, and depending on the patient and its responses against the virus, some of the tests could be more accurate versus some of the tests may not be very accurate. For example, it is well accepted that the RT-PCR kit or RT-PCR test is actually the well tested and validated test. But in same many cases, what happen is that the RT-PCR kit always says the negative, but the rapid antigen test is saying positive and so on. Because the rapid antigen test is actually a delayed test. If you do a rapid antigen test on someone who have and who have got the virus 2 days back, then it is actually not going to give you a test actually, because the test is going to be negative, because antibody development and the proliferation of the B cell and myeloma cells, B cells or plasma cells normally take almost 2 weeks time. So that time, if you have spent and passed, then the rapid antigen test is also going to give you the positive test. Whereas the RT-PCR test is going to tell you about the presence of the viruses into the system. So, rapid antigen test is going to be accurate in the early phase of the disease, whereas the rapid antigen test is actually going to be more accurate in the late phase of the test. So, CT scan is anyway does not give any information about the coronavirus, except that CT scan is actually going to give you the information about the status of the body organs, whether they are healthy or whether they are having any kind of pathophysiology or so on. In fact, CT scan will only say that whether the lungs are actually free of viruses or not, or lungs are free of any kind of inflammation or not. But CT scan doesn't say anything about the coronavirus, because you will probably could see the similar kind of pattern even if you have the influenza virus or other viruses. So apart from the CT scan test, probably the RT-PCR test or the rapid antigen test, both are equally okay, depending on in what context you are actually doing the test. So Sai is asking if I have to isolate a neural receptor which induces apoptosis in Parkinson's disease, which method of separation would be more better to isolate the apoptosis receptor of the nerve cells? Okay, so when you have these kind of questions, or when you have these kinds of question to resolve during your experimental techniques, one of the easiest experimental technique is that if you know the factors which are being received by the receptor, and then it induces the the apoptosis. uh, One of the thing what you have to do is you can actually go with the start from the very basic. For example, whether there are Parkinson disease patients where this apoptosis is not been observed. If at all it happened, then what you can do is you can take the patient who did not show this particular type of apoptotic phenomena versus the patients who have get this kind of phenomena, and then you do a differential display method. Either you can do a microarray test, like you can do a detection of the messenger RNAs at the microarray with the help of the microarray, which are the proteins are differentially expressed in the patient where you are actually observing the apoptosis versus the patient where you are not observing the patients the apoptosis. And if you do a differential display, that can be done even at the protein level as well as the microarray level. When you do at microarray level, it is actually going to tell you the that okay, these 100 genes are not present or not been upregulated or downregulated under the conditions where you are actually getting the apoptosis. Now, once you have that data, then what you can do is you can even do the proteomics experiment, and then proteomics experiment also can be, refined in terms of the what are the factors are being appearing onto the plasma membrane. So you can isolate the plasma membrane and ask about the which proteins are coming into the patients where the apoptosis is happening or not. Now, if you combine these two data together, it is actually going to tell you, okay, these are the 10 receptors or these are the 10 proteins which are responsible for induction of apoptosis in the patients which are having a Parkinson disease. Now since you started with the 1000 or 5000 genes, now you ended up only with 10 genes. Now these 10 genes you can make, either you can clone them and then you ask the same questions whether they are capable of enough or not. Otherwise, alternative is that since you got the 10 genes which are responsible for this kind of phenomena, and out of those 10 genes probably 3 or 4 are the receptors, then what you can do is you can just either do a siRNA technique. You can just knock down some of these proteins and see whether that particular phenomena is happening or not. And if that is not possible because of the ethical reasons or because of the experimental limitations, then what you can do is you can do a ligand. You can take the ligand, prepare the protein from these genes and see which one is fitting into this particular ligand or not. So these are some of the method what you can do where you can actually utilize the different types of high-end techniques, and that actually will probably give you the most conclusive answers about this. If you could not understand any of these aspects, probably you can ask me again and I will happy to help you or happy to discuss and elaborate further. Transcripts are already been done from my side. I have already verified the transcripts and I have asked the IIT Madras to upload the transcripts. But because there is a there could be some technical issues or there could be some delay at their end because you might have know that there are some issues happening at IIT Madras site. So if that can be resolved, I think the transcripts are going to be uploaded very soon and they will be available to you before your exam. And I hope you will be able to prepare well for the exams. So we have only 5 more minutes left. If you have any more queries, you should ask me the questions. Otherwise, we'll wind up this session and then uh, we should no, you will miss the opportunity to ask any query or any confusion what you have right now. And because after this, you're going to have an exam which is coming very soon. Uh, to best of my knowledge, the exam is for 3 hours. But if you want to be very sure, you should ask this question to the IIT Madras people, and they'll be able to help you with that. Okay, so since you all are not having any more questions, I think we are going to wind up this session now. And I'm thankful to all of you that you have attended this course, and I hope you could have get benefited from the some of the content what we have created. And if you have any more queries, future queries or further queries, even if we uh, after the even after the exam also, if you have any queries which may not be even related to this course, you can get back to me and you can write to me or you can be in touch with me and I will be happy to help you. So good luck to for your exams and thank you.